Thanks for listening to the Mind on Fire podcast. Today we're going to listen to an interview that I did with the head of the University of Washington Friday Harbor Labs. Her name is Billie Swalla, and she's got a lot of interesting things to say about the research that they're doing over there. And I hope to talk to her again and maybe ask a lot of follow-up questions. You won't hear me in this one. I'm edited out. But I think you'll enjoy what she has to say. And thank you so much for tuning in. Please subscribe and hit the bell. Thanks.
My name is Billie Swalla, and I'm a professor of biology at the University of Washington. I'm also the interim director at Friday Harbor Labs. And my research is about, I work on the genomics of marine organisms. So marine invertebrates are my specialty. Vertebrates are part of one phylum, and there are actually 38 animal phylums, so most of the animal phylum are invertebrates. I work on at least 3 of those phylums, hemichordates, tunicates, and ctenophores. And I'm interested in body plan. So how does a genome, which we all have a genome inside of the nucleus of our cells, how does that read out to make a certain kind of animal? So how does it make a human, or how does it make, all humans started as one egg. How does it make a worm-like animal? How does it make a sea squirt? And the way that happens is through development. So you have 1 cell, goes 2 cell, 4 cell, 8 cell. And what happens is that there's particular proteins called transcription factors that turn genes on and off. So the first genes that are expressed are transcription factors, and they turn things on and off. And then eventually you get a tissue formed. And of course, we all know that once all the tissues are formed, you end up getting something, a brain, and then that allows behavior and lots of other things to happen. Most marine invertebrates have a larval form, so they'll have a form that's in the plankton that can travel quite long distances. And then they'll go through metamorphosis and have a completely different adult form. So we're also interested in that process. How do they go from a very different larval form to a different adult form? Humans, of course, don't have that. We have the same form that comes through, although some people have suggested puberty is something like a metamorphosis. So there's a very different things happen, of course, at puberty with behavior and bodies, facial features, and et cetera, that you see differently. And of course it's hormonal, and most metamorphosis is due to different kinds of hormones that are being produced that cause the animal to look different. So you might say, well, why would I care about that? Why would anybody pay for that kind of research? One of the animals we work on are hemichordates that are related to echinoderms, but they have a body plan much more like us. They have a nervous system on the dorsal side or the back side, which is exactly what we have. And they can completely regenerate. So you can cut them right in two. So it'd be like if we cut us right at the stomach part, and then you made a new head and a new gut and a new heart and a new kidney, and they make all those new tissues de novo and can make then a whole 2 worms out of 1 worm. And we're very interested in that process. Again, it's a process where you have very defined structures coming from just a little knob of tissue. And so we're very interested in how that happens. And of course, we probably have those same regeneration genes, but for whatever reason, they're not activated in humans. And I'd be very interested in it. Could we activate those and regenerate parts? And one of the things about regeneration is most of the problems that humans have, accidents, spinal accidents, etc., most of the causes are through accidents, and so we're all equally likely to get those. No matter what your lifestyle is, no matter what you do, you're equally liable to get an accident. So that's the kind of practical information that may come out of there. One of the animals that we sequenced here, a ctenophore, it's called a comb jelly, swims around in the plankton, has been very interesting because these swim around in the plankton and are active predators, so they eat things in the open ocean. And it turns out that genomically they look very much like a sponge. So they have many of the same characteristics of number of genes and in developmental pathways. They have the same members, so genomically they look like a sponge, but if you look at them, they look very different than a sponge. So we're very interested in that. It means that our the animal ancestor, which is always thought to be sponge-like, may actually have been an active predator. And so you have everything in between. So it gives us interesting insights into evolution as well as what's going on in the genome. The other really interesting thing about the ctenophore genome is that they have duplicated a group of genes called the glutamate receptors. And we have glutamate receptors. We have about 12 of them that we use in our cortex. So we use them for higher thinking functions and et cetera. Whereas they've multiplied these, and it's their only, as far as we can tell, it's one of their only neurotransmitters. They don't have serotonin or dopamine or many of the other neurotransmitters that we have. So it looks like it's almost an independent evolution of a nervous system. And that's a really interesting, insightful thing, too, for how do you evolve nervous systems. And, of course, it may help us understand the function of these different receptors as well. So that's another sort of practical kind of thing that might come out of this research. When we do these studies, I do them both within my lab, and then we also have courses called research apprenticeship. So I have 5 or 6 undergraduates, sometimes 7 or 8, up to 10 undergraduates that work with me and another faculty member, and we have them do everything from catch the ctenophores to extract DNA to looking in databases and doing bioinformatics with what's known about the genes that we have. This is a great, tremendous opportunity for them. They get to learn all about genes and genomes and spend a quarter out here, and it helps them figure out whether they like research. We've been able to get students really excited about research and going on to graduate student school that way. And so we're very excited about that research apprenticeship program. So I always say that one of the best things about my job are the people that I work with. So working in academics, you meet lots of people, all the way from faculty to young students who aren't sure what they want to do. Sometimes not very focused and looking for something, for what to do with their life. And so it's really fun to work with them. And, many times we change lives. And my life was changed, as I said, by going to marine biology lab. So I feel like I'm giving back to that whole process for students. At the lab, we have an ocean acidification environmental lab. So we have Emily Carrington's one of our faculty who's working on ocean acidification. There's also a lot of ocean acidification work going on campus. So the Friday Harbor Labs is part of the College of the Environment. And in the College of Environment, there's a number of different divisions, including fisheries, oceanography, earth and space science, Sea Grant. There's a number of there's about 12 different divisions in the College of the Environment. And so ocean acidification is one of the big things that we've been looking at here. It turns out along the Pacific coast, because of upwelling, sometimes the acidification is very high and sometimes it's lower. So one of the things we do is monitor temperature and ocean acidification, and then also study the effects on embryos. It turns out it makes muscle byssal threads weaker. There's a lot of different things that ocean acidification does. We have Jan Newton and Brett Byers. Jan is from APL, the Applied Physics Lab on campus. They've been doing a pelagic ecosystem function, which they've been doing continuously for over 10 years now. So that tells us what things are in the plankton, in the water out here, in the San Juan Islands, and they do the same transects year after year. So we're starting to see some long-term trends as far as what's going on, fish larvae, algae, everything that they get out of the plankton. We also have Ken Stevens is doing some long-term ecological studies. So he has transects. This is a marine preserve, actually, around where the labs owns the property. We also have marine reserves, and Ken Siebens has some long-term plots where he's looking at what happens. He goes out every month or something for however many years and looks at the same plot and looks at how it changes over time. So if there are long-term changes that are beginning, we'll be able to pick those up. Adam Summers is interested in fish biomechanics, and Adam does lots of really interesting things, but one of the things he does is look at fish skeletons, and right now he has an art exhibit at the Seattle Aquarium on some very, very beautiful fish skeletons that he's obtained to use for his research, but you'll see that they're also really, really gorgeous skeletons. We also have Richard Strathman is an emeritus professor. He was the associate director for many years, and he works on larval biology, and so this is Friday Harbor Labs has traditionally been a place where a lot of very important larval biologists got their start, and there's still a larval biology class that we have that he continues to do. So we try to do the range from all from oceanography, open ocean type things, to fisheries, to sort of marine invertebrates, and looking at both ecology, biomechanics, genomics, lots of difference. We have people working on different things, but also looking at different levels on things. And in the summertime, we have all kinds of people here. I've started a new series called Tide Bites that I've been sending out once a month to let people know the research that we're doing. And we're starting with the research of people here on campus, but I'm hoping in the summertime to start to get our visitors to do some of them so that we know their research as well. I realized this last summer because some people had come and left, and one of the researchers ended up following up and sending back to me what they had found in the summer and how exciting it was, and they were looking at a very unusual sea anemone Stampia that has an escape response, but they found another behavior that it does, and it helped them understand again neural function and how you can have nerves do different behaviors. And so it's fascinating, and I realize I don't get to see that with most researchers. They come and go, and unless they send us back publications, we're not really we know that they're doing cool stuff, but we're not sure what that is. And so I'd like to get more of them also talking about their research and getting their research out there. One of the things I've been trying to do as director is get out to the people of San Juan Island, all of the San Juan Islands in the state of Washington, the things we're doing up here, because I consider this a research institution, but I was telling one of our advancement board members this summer, it's a research institution. She said, well, I don't know what research you do. And she said, we understand teaching, we understand the class functions, but we don't understand the research. And which is why I started Tide Bites. And it was it was really good feedback, because I've been getting lots of feedback from people, even people in our department, saying that they love reading them, that they love hearing what's going on, that they're very excited to see the research happening and to see us getting the word out, and that they're really enjoying seeing what's going on here. So I'm very happy about that. We also have an open house this year. The open house is going to be on May 11th, which is a Saturday, where anyone can come and see the labs. We have free popcorn. You can buy hot dogs and chips and soda. And in years past, we've had a marimba band playing, which was a lot of fun. And basically, most of the researchers are here. People come, and the classes that are going on will have things set up so you can go through and see what they're doing in a particular class. And so we would love to get a huge turnout for that. We've had up to 700 people here on campus during those days to, again, kind of get a hands-on about what we're doing. So we really think of ourselves as being interested in ocean health. So that's kind of the term people are using now. So we want a healthy ocean. The San Juans have a very, very diverse number of animals that live here. That's because water comes in on either side of Vancouver Island and larvae swirl around here and settle out. So we have a higher diversity here than even on the outside of Vancouver Island or on the coast of Washington. There's also a lot of habitats. Because there's a big tidal exchange, we tend not to get pollution or sulfuric places, which is not the way it happens in Hood Canal. If Hood Canal doesn't get the water exchange, sometimes they go through these very anoxic periods and almost everything dies. Because we have high water exchange, we tend to have pretty healthy environment here anyway. One of the things that actually Megan Detier is working on here is when people put concrete out on their shore to try to preserve the shore, that actually kills a lot of the animals that are on the shore. So one of the things that she's been doing is working with parks and et cetera to take out any sort of concrete pilings or whatever, because it turns out that that exchange between the water and the shore is really important and very important for fish larvae and for lots of animals. Their larvae or eggs live in the sand, and so they need that habitat to be able to be healthy. So this is one of the things that we're trying to weigh in on. Also, the importance of reserves and preserves. So there is a push to try to get the west side of the island put into a preserve. Part of that's for the whales, for the orcas, so that there's salmon there for them to eat. It's also would be really important because there are sea urchin fisheries and sea cucumber fisheries that have opened back up, and they will take huge amounts of sea urchins and sea cucumbers. And what happens, the abalone right now are to this level where if they fall to below a certain level where the eggs and sperm can't reach each other when they're spawning, then you'll actually have no more abalone. So the abalone now are almost extinct here in the San Juans, which is really sad, and we don't want to reach that level with any of our other animals. So we are looking very carefully to see what's going on. The ocean acidification has been something that's increased in the past 10 years since I've been here because the increase in CO2 in the atmosphere. So as the CO2 increases in the atmosphere, that causes a reaction with the ocean where you end up getting this acidification. And even changing a few decimal points, so a 0.1 or 0.2 change in ocean pH can critically affect the animals that live there. And that's one of the things that we're studying. So that's become a problem. As I said, we're starting to understand more that on the Pacific coast, as there's upwelling events, which you have in the summertime, those upwellings bring even more acidic water up. And so you have what some people have called ocean acidification storms. So there'll be 2 or 3 days where it's very highly acidic. We're just starting to understand this because, and it's important because if you're doing an experiment at that time, you might get some result. And then if you're doing it at another time when the pH is different, you might get a different result. So that's why we're trying to monitor that and pay attention to what's going on. We're lucky here in the San Juan Islands. There's not much pollution. People are very civic-minded. If you go out on the beaches, there's very little trash on our beaches. If you've ever been in Florida and walked the beaches, you'll see what I'm talking about. I used to take a bucket when I was out there collecting and fill them full of stuff. But this ocean acidification and the CO2 rise and everything rising sea level are all very, very valid concerns. The rising sea level, what's interesting is that the San Juan Islands, because we are near the Juan de Fuca Fault, which means that the San Juan Islands are also rising at about the same level as the sea level is rising. So there's not that immediate concern for the San Juan Islands, although in Seattle and Puget Sound, I think that those are valid concerns, especially for people who have their houses, of course, very close to the shoreline. So we're doing well here, but we're very vigilant and, of course, always keeping our eye out to see what's going on in other areas.